Okay, so this week's lecture is going to focus on cell structure. Here's a list of the learning objectives for this section. The first part, we're going to start by talking about microscopy. Although this isn't a direct course competency, if you don't know how a microscope works, you're not going to be able to do a majority of the labs that we do, and you're not going to understand how we actually visualize cells. So we'll start by doing just a brief overview of microscopy. So the light microscope is the microscope that we use in this class. It uses visible light for observing objects, and the one that we use specifically is called a bright field microscope. And this is the most common that you'd find in a science lab. If you're going to purchase a microscope for yourself at home, it's going to be a bright field microscope. Now what bright field means is that the field, or the background of the specimen that you're looking at, is bright, while the actual specimen appears dark. So when you're looking at it, the specimen looks darker than the background. So it's a bright field, a darker specimen. There's all different types of microscopes that exist, and if you go on to take a microbiology class, you will definitely get into those different types. But for this class, we'll just stick with the bright field microscope. In order to understand magnification, we have to look at some of the parts of the microscope. So if we look here, you can see here are the objectives, and the objectives are 4, 10, 40, and 100. Now light is going to come up through the bottom of the microscope, through this condenser lens, which focuses light through the specimen. And then the first place that it gets magnified is here in the objective lens. So the objective lens is again 4, 10, 40, or 100. Then the image is going to be bent and it's going to go through these ocular or eyepieces, and there's a lens in here as well that's going to magnify it again. So the second place that it gets magnified is in the ocular lens. Most standard ocular lenses run at about 10x, so they multiply the image 10 times bigger than what it actually is, while the objective either multiplies it 4, 10, 40, or 100. In order to get total magnification, you would multiply the objective lens times the ocular. So if we're looking through the 40x objective, the total magnification would be 400 times, because you would do the objective times 10. This is an overview of the different parts of the microscope. Take some time to familiarize yourself with these parts, and there's a couple videos that are posted on the Blackboard site that go through how to use a microscope, the different parts of the microscope, and so forth. So this is what we're looking at here, all the different parts. You've got the eyepieces, also known as the ocular pieces. You've got the arm and the base, which is how you would transport the microscope. One hand goes on the arm, one hand goes on the base. And then you have the adjustment knobs that allow you to focus the microscope. So the coarse adjustment is really only used for the 4x and 10x, while this fine adjustment is used for the 40x and 100x, typically. Then you have these knobs here. These are stage adjustment knobs. So when you put your microscope slide in here, you're going to put it right in here, and this is kind of a little switch or a little lever that's going to hold the microscope slide in place. The old microscopes, you used to actually slide them underneath the stage clips. Now you just kind of hold it in place using these levers here. But in order to centralize the specimen that you're trying to look at on the microscope slide underneath the objective, that's when these stage adjustment knobs come into place. So one of them is going to move it left and right, the other one moves it back and forth so that you can center it underneath the objective lens. Then we have the light adjustment knob. This is another light intensity control. So this diaphragm kind of controls how much light is allowed up through, and then this controls the brightness of the light. Now down here is the condenser, and the condenser also has a diaphragm control on it. Once we start to look at cells under the microscope, you should definitely play around with this little lever here, this diaphragm control, because it allows you to see different depths within the cell and different details. So if you make it a little bit darker, you can see different details on the inside of the cell. So it's really cool to mess around with that. Then we have, again, the objectives, which are anchored on this nose piece part. So the nose piece holds all of those objective lenses. So how does a light microscope work? Well, light is focused on the specimen by the condenser lens. It passes through the specimen. The image enters into the objective lens where it's magnified, and then it enters into the ocular lens where it's magnified again. This one is key. Light passes through the specimen. So this kind of limits the thickness of the specimen that we can actually put on a microscope slide. It has to be thin enough so that light can pass through it. Now we have this 100x objective, also known as the oil immersion lens. So oil immersion is the 100x objective. At this high magnification, there is a lot of interference with light scatter. So under normal circumstances, light is going to go up into the objective. Some of the light is going to get scattered away from the objective. If the light is scattered away from the objective as pictured here, this is not going to come into focus. So how do we force all of the light up into the lens so that we can have better resolution and we reduce refraction? Well, we add oil to the surface of the actual microscope slide. So this is the slide, the microscope slide that's sitting on the stage. This is your objective lens. And you see the oil drop that's been placed here actually forms a film between the lens and the top of the microscope slide. If this film is broken, it doesn't work. Then light's going to go back to scattering all over the place. So the oil kind of forces the light to go up into the objective to bring this objective lens into focus. This one will not come into focus for you unless it is immersed in oil, which is why we call it the oil immersion lens. We will use this to see the very tiny microorganisms. So bacteria and fungi are often used, or we use that 100x objective for.